Hi, how are you?
Good, how are you?
Good, thank you. Thank you for coming.
Yeah, of course. Um, okay, so it's up there, and then anybody can stream through it. I have it up on my computer.
Yep.
Okay.
So yeah, everyone can go through it. So yeah, it's really helpful to say like when you skip slides that to just say on which slide you are on.
Okay.
We'll start in around 5 minutes.
Okay.
So, but yeah, I hope you had a great day until now.
Thanks. Yeah, no, it's good. How are you? How was your day?
Well, I'm actually moving tomorrow. So
Oh no.
I went to work and now I was packing. So yeah, but I'm good. It's a good move. So we are moving to apartment with our own backyard now in the city. So I'm really actually happy. So
Oh, that's nice. It's probably pretty rare to find there.
it's so lucky. So it's actually a new building right on the same street. So the kids are already used to this neighborhood and people know them.
That's great.
But
Yeah.
But I kind of felt, I'm just moving. We are just moving next door, so I kind of didn't do as much. So now I'm in trouble. Basically,
Oh.
the movers are coming tomorrow, and I didn't do everything.
Oh. Well, hopefully you can pack well. Listen. Yeah, we're also moving in 2 weeks, but we're going from a smaller house to a bigger, so we have less stuff. It hopefully won't be too bad, but we'll see.
Yeah, that's nice. So you guys bought a house?
Yeah, so we're kind of like you, same neighborhood, just 2 blocks over, but
Oh,
a
okay,
little bit bigger room.
that's nice.
Yeah.
So our rooms will
So,
actually be smaller, but we'll have a backyard. Like it's the same amount of rooms, but they
yeah,
are smaller. But we'll have a backyard, which I like for the kids. So
yeah, definitely.
anyhow. Yeah, the schools are really good where they are, and they can, my son can walk there, and so I kind of want to stay around here. So
Yeah. How old are they?
so my older son is already, grown up and moved out, but then I
Oh,
have a 12-year-old and an 8-year-old
oh
daughter.
wow. Yeah.
Yeah, I was really young when I had my first son. So.
But those are fun. Well, somewhat fun ages. I've heard my nephew and niece are 11 and 12. We're gonna go see them this weekend. They're still fun. They're not yet mean.
Yeah, right. It's the last moments when they are still like humans and then they are monsters. I know. Like my 8-year-old, she's still really so nice and stuff and gets happy
Aw,
about like stuff, you know.
yeah.
Will change
Cool.
soon.
Hopefully not too soon.
And it's all on record. No, because this is recorded. My kids will.
Oh, they'll forgive you. Sorry, I forgot about that.
Yeah, me too. And I should know. Like, I don't know. Everyone is kind of late today. But we still have a few minutes to start. Hi, Serena.
Hello.
How are you?
Hi. Good.
Yeah, we'll start in a minute, if that's okay. Um, yeah. Thank you, everyone, for coming. We're almost starting. So,
Hello, Dr. Zuniga. Is that how I say that?
Yeah, that's right.
Oh, I'm missing the flags. How do you get the flags?
so you put them somewhere in your bio, and then it appears right now for you know this. It like on some occasions this works.
What do you have to put in your bio?
The flag, like the rainbow flag, and then it should be
Oh,
working.
okay.
So, hi, Dr. Mariam. How are you today?
Hi, Katerina. Hi, Serena. And hello, Dr. Zuniga. Welcome.
Hello.
Hey, how
Yeah,
are you?
yeah, I'm fine, thanks. I'm good, thanks, Katerina. Hope you all are doing well.
Yeah, I think we can slowly start by introducing you. How Eli is coming. Hi, Eli. How are you today?
Hey, pretty good. It's funny. Cami was talking just in the Ukraine room about an article she's got coming out on fluorescence microscopy, AI analysis of it related to tau. So that will be coming out probably on her blog tomorrow, and I'm certainly looking forward to it. And I thought that you might be as well, so that's why I mention it.
Oh, cool. Thank you. Okay. Welcome everyone to the Science Society. Thank you all for coming, and special thanks to our guest speaker here today, Gabrielle Zuniga. And let me give you a little bit of introduction about her. Gabrielle Zuniga is a MD, PhD track in the Department of Pharmacology. On the for the PhD part, she's on the neuroscience track at the University of Texas Health, San Antonio, and she did her bachelor in science and biology with honors, and she did a concentration in neurobiology. And then at the University of Texas Health, she did an integrated biomedical sciences graduate program, PhD neuroscience track. And she's also in the School of Medicine because she's doing MD, PhD at the Texas Health San Antonio. She received various scholarships, so already in her early career, she's very accomplished, and received also awards and honors in Tau Consortium for a Tau Consortium Fellow, and for her different presentations, she got awards. So yeah, and many more. It's a long list, and she has clinical experience already and paper and published papers. So I don't know how you did that. Did you ever sleep? Welcome.
Thank you for that introduction. Yeah, I and thank you for inviting me to talk about our paper. So this is our first author, or my first author publication in Dr. Frost's lab here at UT Health San Antonio, and just defended a couple of weeks ago, and so I will be transitioning back to med school. So halfway there, or more than halfway there. But yeah, if there aren't any questions, I can jump into the talk. Or
Yeah, usually we ask like a general question. I don't know, Serena, do you want to go ahead?
I'll do it. Yeah, okay, so welcome. We'd like to ask a general question for the benefit of the audience and get to know you a little better. Can you think back to a time, could have been early in life, where you really knew that science was going to be your thing and you had a special connection to science, and could you tell us about that?
yeah, it really wasn't super early on. So I was raised in Chapel Hill, North Carolina, which is basically on the University of North Carolina campus. My dad is a oral maxillofacial surgeon, also a PhD in neuroscience. So I grew up around academia and medicine and knew that medicine was what I intended to do. But it really wasn't until I went to UT Austin, and they have these great research programs for freshmen and first year students to really kind of just explore whether or not they like science or not. So the vast majority do 6 months or a semester and decide that's not for them. But there's a subset of us that stayed in lab late, or really brought our projects that were really just meant for a couple months past that point. And I think it was really just coming back and realizing that, I really enjoy this, and spending more time in the lab than classes really solidified that for me.
Well, that's great. So you kind of knew it, you had to do something in medicine and science, but you sort of found your way, which brings me to your next question, or my next. What was the specific path and decision that brought you to the work that you're going to present today?
Yeah. So I ended up joining a lab with Dr. John Pierce Shimamura at UT Austin, and his lab uses C. elegans, so the nematode model system, to study Alzheimer's disease pathways and Down syndrome. And so I got involved in a project that was looking at a different target, but looking at what happens, similar to what we do in this lab, what happens when you genetically modify a target, and then what happens to the neurons. And what's super cool about the worms is that you can see everything. So I was able to score the living worms, their neurons, their synapses, and decay. So I thought that work was really, really cool. And luckily, Dr. Frost was interviewing at UT Austin, and my PI really, really thought she was great. And when he found out that I was going to the MD PhD program at UT Health San Antonio, and so was she, we linked up, and I really was excited about her, her approach to answering the question about different pathways in Alzheimer's disease and focusing on tau specifically. And in the lab I was in undergrad, we actually focused on amyloid beta. So it was a nice switch to kind of look at a different perspective in a very, very, very complicated disease. That kind of led me here and in the work that we just published.
Well, wonderful. So the audience can follow along on your slides. They're linked at the
Yes.
top. And so you're welcome to take us away. The stage is yours.
Awesome. Well, thank you guys all for coming. Hopefully you're a little bit familiar with the paper, or you're able to see it, but I'm going to go through the slides, and then we're going to start by talking a little bit about the history and background of Alzheimer's disease and related tauopathies, since this is a little bit more of a broader audience. And as I kind of alluded to, I want to get into how our lab approaches the study of tauopathy, and then the identification of novel targets with disease-modifying potential. So if you start on the first slide, so this would be slide 2, just below the title slide. For those who don't know, it's now been, unfortunately, well over a century since 1901, when Dr. Alzheimer was a clinical psychologist at the Frankfurt Hospital in Germany, where he was also the attending physician for Augusta Dieter here. She was a 51-year-old female who was presenting with quite striking symptoms, including reduced comprehension and memory, disorientation, paranoia. She had this loss of ability to understand and express speech, unpredictable behavior, and pronounced psychosocial impairment. So it was for the next 5 years that Dr. Alzheimer would follow her symptoms and treatment, and it was upon her death in 1906 when he was able to analyze her postmortem brain tissue samples. He used the Bachelski silver stain, and luckily his colleague, Dr. Niesel, he used those stains that he developed as well. And what he found was quite remarkable and really unlike anything people had seen before when associated with the clinical symptoms that he observed by Augusta. And he described these histopathological findings, which are known now as senile plaques, which we know are composed of amyloid beta, and these neurofibrillary tangles. So below the picture of Dr. Alzheimer and his colleagues are these sketches that were in his notebook. And they're these teardrop-shaped tangles that are found inside neurons. And so, while other physicians also observed and similarly reported these neurofibrillary tangles and amyloid plaques, the eponym Alzheimer's disease came to be because it was published by his colleague, Emil Kraepelin, in the 8th edition of the Handbook of Psychiatry in 1910. So became Alzheimer's disease as we knew it. If you scroll to the next slide, I want to talk a little bit more about how, so all that happened in the early 1900s. However, it wasn't really until the early 1980s that tau was found to be the primary component of the neurofibrillary tangles. So in the bottom image, I show a microtubule and these representative tau protein, which normally are preferentially localized in axons, and they stabilize microtubules and prevent them from being destabilized. However, under pathological conditions, tau becomes mislocalized, and it can form these fibrillar tangles, and it becomes phosphorylated. And so, although tau is infamous for its role in Alzheimer's disease, it's really important to note that Alzheimer's disease is just one of 20 plus tauopathies, which is just a general term that we give to describe a group of neurodegenerative disorders that are characterized by these inclusions containing abnormally phosphorylated tau in the brain. So what led us to the lab and Dr. Frost to focus on tau is that we now know that it forms or accumulates in the brain decades before symptom onset. So before the patients show up and are clinically diagnosed, as shown in this kind of upper image, where you have a nice normal brain, you're still going to have abnormal tau deposition. This is way before patients show up to clinic. And in some cases, it's actually true that abnormal tau accumulates before these senile plaques, which are also shown in that image, suggesting that maybe it's more of an initiator protein than the amyloid pathway hypothesizes. It's also true, if you scroll down to the next image, that the tau spread through, the next slide 4, that tau spread through the brain from beginning in the entorhinal and transentorhinal cortex, then to the hippocampus, and finally the isocortical area, that this spread is the strongest correlate of clinical symptoms in patients with Alzheimer's disease, suggesting that tau may be a good therapeutic target for patients with Alzheimer's disease and related tauopathies. So, to give you some background on the history of our attempts to treat Alzheimer's disease in other patients with related tauopathies, it's been almost 2 decades since 2003, which was when the last drug was approved for symptomatic treatment of Alzheimer's disease. And since that time, there's been over 200 interventional therapies that have made it to the clinic. In the left, graph showing the different kind of agents by mechanism on the outside and by phases, phase 1 on the outside, phase 2 in the middle, and phase 3 in the center. These are all clinical trials, agents that were in clinical trials in 2021. So they do a review every year. In 2022, review by Cummings. And in an outline, I think these are the tau targeting. Yeah, there were 13 tau targeting agents in 2021. However, since 2003, excuse me, there have been, of these clinical trials, 25% have directly targeted the amyloid pathway, whereas only 12% targeted tau. And all but 1, maybe 2 of the 50 agents that are directly targeting amyloid beta, they all failed, except for those 1 or 2. Whereas for tau, there's been over 20 targeting tau, and 7 have failed. And that one in the right circle is showing those that failed in phase 2 and phase 3, showing that the majority do at that later stage. And then I want to finish this introduction by pointing out now in the new graph in color on the right. So these are the different agents that were in clinical trials in 2021, and then they're just showing the different phases by color. And so there are several agents that target tau and amyloid specifically, but of these disease-modifying agents, some of them don't. So these in our lab and other labs that have kind of motivated these trials have a different idea and a different approach. So we think that rather than targeting the initiating proteins tau and amyloid that we know are, accumulating in the brain of affected individuals long before symptoms, we want to target mechanisms downstream of pathogenic forms of tau that are well connected to neuronal death. And if you go to, this is slide 7. The way our lab approaches this question of, what mechanisms are downstream of tau-induced neurodegeneration specifically, we use a Drosophila model system. So I went from C. elegans to Drosophila. And the Drosophila model of tauopathy was developed in 2001 in the Feeney lab. And this model expresses a disease-associated mutant form of human tau, the tau-R46W mutation. And as you can see, it recapitulates key features of the human disease. So I'm just pointing out 2 briefly. It shows progressive neurodegeneration. So at day 20, if you look at the fly brain, in this figure, it has a D on it, you'll see vacuoles, pretty large vacuoles. And in Drosophila, vacuoles are very much associated with neurodegeneration. And so when they quantified the number of degenerating neurons with age, they found this really nice progressive age-related neurodegeneration, especially in the R46W. But what was really cool is it also occurred in flies that were panneuronally expressing just wild type non-mutant human tau. So these flies are more of a model of sporadic tauopathy, because patients with late onset sporadic Alzheimer's disease will accumulate wild type forms of human tau. So the degeneration is less toxic than the R46W, which I'll kind of mention a little bit later too. They also have a shorter lifespan, so flies can live, they can honestly, control flies can live up to 100 days. However, the tau flies, they get really, really sick around 30 days. So unless I mention otherwise, for the majority of studies, we use day 10 old tau transgenic Drosophila, because this is a really good, they show moderate levels of neurodegeneration, so we can detect enhancers or suppressors, just different kind of genetic modifiers. And then this is prior to this really exponential decline in survival. So using these Drosophila, the basis for this paper was first found by Dr. Frost in her 2016 paper, where she found that in these flies, if you look at the neuron using an antibody that recognizes lamin, which is shown in the left figure, this is slide 8. In the left figure in green is lamin. It's an intermediate nuclear filament protein that underlies the inner nuclear membrane and provides anchoring of highly condensed heterochromatic DNA. So it really provides the three-dimensional architecture to the genome. And using this antibody, she found that in control, you have nice round nuclei, whereas in tau transgenic Drosophila, you have these weird circular, what we call invaginations of the nuclear membrane. And these are specifically in neurons. And this is way more, if you go to slide 9, just below, in humans, using the same using an antibody against lamin B, you also see this, but there are more of these like finger-like or tunnel-like projections into the interior of the nucleus. And when she co-stained this with an antibody that recognizes nuclear pores, we found that these invaginations are lined with nuclear pores, suggesting, as in the model to the right, that in tauopathy, you have both the inner and outer nuclear membrane invaginating and a cytoplasmic core. So perhaps we thought that different molecules would be being shuttled between the nucleus and the cytoplasm, including RNA, protein, and DNA. And we chose to focus on RNA. So if you go to the slide down, slide 10, I show an image of the central dogma, which basically says that genetic information flows from DNA to messenger RNA via transcription, and from messenger RNA to protein via translation. And in eukaryotes, you have the nuclear envelope separating transcription in the nucleus from translation in the cytoplasm. And messenger RNA is the molecule that stores and transfers genetic information between the two compartments. So it's really this really critical molecule in this transfer of genetic information, the fidelity of gene expression. And if you go to slide 11, I'm just pointing out that mRNA steady states are really determined by this ratio between synthesis and degradation. However, if people have shown this in different model systems, different cell types, that mRNA translation rates rather than the mRNA steady state levels are more predictive of protein expression, suggesting that what happens to the RNA after synthesis are the processes that are much more important in predicting protein production and, ultimately the phenotype. So the observable characteristics such as high eye color and blood type. So it's really critical what happens to RNA after. And we focus on the role of the nuclear invaginations in this. So if you go to slide 12, there's data, explaining what happens to RNA after. So when the RNA is synthesized, it must travel from its site of transcription to nuclear pores within the nuclear envelope. And for example, I use the nucleolus, which is a site of our RNA transcription. And so normally, there are kinetic studies suggesting that the rate limiting step of mRNA nucleocytoplasmic transport is the diffusion distance from this site of transcription to the nuclear pore. And so we thought perhaps if you go to slide 13, that a decrease in this diffusion distance, which is mediated by these nuclear invaginations, could have a positive effect on mRNA export rates. So there was a postdoc in our lab a couple of years ago who published on this in Aging Cell, in which he looked at the relationship between poly-A RNA and these nuclear invaginations in tau-transgenic Drosophila flies heads. And using a poly-DT probe that recognizes poly-A RNA, if you focus on the top nuclei with the arrowhead, he found that poly-A RNA frequently accumulates within these invaginations, suggesting that possibly RNA was being exported through these invaginations and accumulating there. Whether it was stuck or whether it was just massive amounts of RNA, we're not quite sure, but the paper kind of suggests that. And so he saw this relationship. He wanted to know if this mattered for neuronal health in the tau-transgenic Drosophila model. And so he genetically manipulated SBR and NXT1, which are Drosophila homologs of the nuclear export factors in humans. And he found that if you suppress SBR and NXT1, reduce their expression level, that this suppresses tau-induced neurodegeneration. So that suggested to us that perhaps an increase in mRNA export would enhance tau-induced neurodegeneration. But ultimately, this suggested that aberrant nucleocytoplasmic export is a causal mediator of tau-induced neurodegeneration. So all of that introduction to kind of bring you to our thought process and what led to the basis of this paper. He showed that there's excess RNA export and that it manipulates tau-induced neurodegeneration, but why is this toxic in the context of tauopathy? Normally cells have multiple mechanisms to get rid of excess RNA or just bad RNA in general. There are a whole set of RNA quality control mechanisms, so why in the context of tauopathies is not working properly? We thought that tau-mediated increases in mRNA export lead to neuronal death by essentially overwhelming the RNA quality machinery and limiting its ability to clear aberrant RNA transcripts. So to do this, we focused on nonsense-mediated RNA decay. It's a highly conserved RNA surveillance mechanism that is probably the most well studied of the RNA quality control mechanisms. In humans in Drosophila, it's predicted to affect about 10% of the transcriptome. Based on its name, it was historically thought to target just transcripts containing premature termination codons. But it's now very well established that it also targets normal, quote unquote, normal transcripts. So on slide 17, I'm showing a quote unquote normal transcript with an intron in the 3' UTR. So this is a NMD triggering feature. And so this it's an NMD reporter transgene that was previously established in Drosophila by the Metzstein group to be targeted and degraded by NMD. And so when they knock down NMD core target core factors, and they used the C. elegans model to establish this, that it significantly enhanced fluorescence of this NMD reporter, which led to more red fluorescent protein. And so I'm highlighting in this cartoon here that when a ribosome hits this normal termination codon, the core factors UPF1, UPF2, and UPF3 will essentially assemble onto the transcript and lead to a cascade that then results in recruitment of SMG5 and other SMG factors that will cleave the transcript and lead to degradation. So in normal cases, when everything's working fine, you should see low levels or low expression of this red fluorescent protein. However, when we set out to first determine if NMD is blunted in tau-transgenic Drosophila, we found that this reporter protein not only is significantly elevated in Drosophila that have reduced NMD activity by reduced expression of SMG5, they also have reduced enhanced red fluorescent protein expression at day 1 and day 10 old tau-transgenic Drosophila. So what's really cool about this is that, as I kind of mentioned, day 10 we see moderate levels of neurodegeneration. But at day 1, you don't see you don't see normally any neurodegeneration, and we really don't see any other other tau related mechanisms taking place this early. So this was really interesting to see that NMD sensitive transcripts are not only stabilized at the RNA level, but they're also stabilized at the protein level prior to neurodegeneration. So this deficit in NMD seems to be an early event. And then if you look at slide 19, to cover our bases, we also wanted to use an alternate approach to measure NMD activity. We decided to look at the steady-state RNA levels of GAD45 and ARC1, which are known targets of NMD and Drosophila. And we validated that here using a RNAi expression line of SMG5. So in controls, you get low levels of GAD45 and ARC1, whereas in SMG RNAi Drosophila, you have increased expression. So these aren't being appropriately cleared. And then same thing when we looked at tau-transgenic Drosophila heads. So again, really cool was we saw this at day 1 and day 10, that both expression of GAD45 and ARC1 were being increased, and these transcripts don't have premature termination codons, so these are quote-unquote normal RNA that are decreased clearance in tauopathy. So we see blunted NMD activity in tau-transgenic Drosophila with the mutant form of human tau. What happens in Drosophila model with expression of the human wild type tau? Pretty interestingly, we're never super sure. Usually we see, like I suggested, a little bit moderate level of toxicity compared to R486W. But even in this model, there was reduced NMD activity, both using the NMD fluorescent reporter protein as well as the GAD45 and ARC1 steady state levels. So this reduced clearance of RNA by NMD seems to be a conserved feature of tauopathy. But similar to what Garrett was asking, does this really matter? Do tau-induced deficits in NMD contribute to neurodegeneration? So, moving on to slide 23. Let's see. I moved that one. Moved to slide 24. I must have moved those around. Using a similar approach, we used tunnel which I did not mention earlier. It is a marker of double-stranded DNA fragmentation that's associated with apoptosis. And we found that when you reduce expression of UPF1, UPF3, and SMG5, or overexpress a dominant negative mutant of UPF2, that this significantly enhanced tau-induced neurodegeneration. And then when we did the opposite and we overexpressed NMD core factors, this significantly suppressed tau-induced neurodegeneration. So while it was really cool to see that when we broke this pathway that it enhanced tau-induced neurodegeneration, it was even more exciting to see that when we try to fix it, it worked by overexpressing NMD core factors. So seems that increased clearance of RNA by NMD suppresses tau-induced neurodegeneration and is essentially neuroprotective in tauopathy. And then slide 25, we included this to show that, this wasn't just simply by our genetic manipulations targeting tau. So tau levels were the same in each Drosophila mutant, and the effects we were seeing were not just simply because tau was being affected. It wasn't messing with the model, so that was good. We showed that NMD activity is blunted in different models of Drosophila models of tauopathy, and that it does matter. It does affect neurodegeneration. Now, we wanted to know if there was, similar to the poly-RNA, is there a potential link between blunted NMD and these tau-induced invaginations? And so we first thought that, maybe the invaginations were forming these physical barriers. Sorry, just wanted to check. Oh, shoot. Sorry. Okay, sorry, I think I lost. So yeah, we wanted to see if these invaginations were forming physical barriers that were essentially keeping out NMD targets, core factory, and machinery, and sequestering the poly-RNA within the invaginations. And so we used fluorescent in situ hybridization combined with immunofluorescence using the poly-DT probe, again, to recognize poly-RNA. And we found, if you move to slide 28, that very clearly within these invaginations are both the poly-DT and UPF1, which is an NMD core factor. So this suggested that the intravaginated RNA was physically accessible to UPF1. So it, at least in this case, was not acting as a physical barrier, the invagination. However, when I looked through the brains completely, I also frequently found cases where it was just poly-RNA and not the UPF1. So, and if you move to slide 30, I'm just showing up close because it's hard to see it from a 2D image and not really appreciate that it's fully within these invaginations. And when I quantified this, it seemed that, about 2/3 of the poly-enriched invaginations lack UPF1. So while it's not, the invagination is not necessarily a physical barrier, it seems that most RNA containing nuclear invaginations are not actively clearing RNA, at least via NMD. So there are a couple possibilities. So moving to slide 32, the RNA could simply just not be an NMD target. Maybe it's just normal RNA that's freely being expressed like it should. And so we wanted to know if, or that it's a physical barrier, but at least in some cases. So we wanted to know what percentage of these, or if any of these RNA are quote unquote bad RNA that are targeted by NMD. So using the NMD fluorescent reporter protein, I wanted to see, are these do these accumulate, does the protein accumulate within the invagination, and are they being effectively cleared? So I was able to use, this is all immunofluorescence, so we're looking at the protein here. And I did see accumulation of the NMD reporter as shown in slide 32, clearly within these invaginations, shown in slide 33. And when we quantified that, I think it was about 40% of the invaginations are enriched for this this red fluorescent protein within the invagination. So that suggested that the RNA within these invaginations are supposed to be cleared by NMD, but they're not, at least not in the context of tauopathy. So if you move to slide 34, we wanted to know if this RNA was not being appropriately cleared by NMD because, thinking back to our hypothesis, because maybe there's just too much RNA compared to NMD machinery. So slide 34, we just, reviewers wanted us to validate that not only was genetic overexpression of UPF1 leading to more UPF1 at the RNA level, but it was also leading to more protein of UPF1. So it seems that the effects that we're seeing is because there's more UPF1 activity and more NMD activity as a consequence. So when we had this more NMD activity and UPF1 overexpressing tau-transgenic Drosophila, you got reduced NMD fluorescent reporter protein expression, as well as reduced expression of GAD45 at the RNA level. Well, there seems to be somewhat of a trend for ARC1. It didn't seem to affect those levels as well, for whatever reason. And importantly, though, if you move to slide 36, as I showed before, in tau-transgenic Drosophila, poly-A RNA accumulate very clearly in slide 37 within the invagination. And I love that nuclei because it has multiple invaginations right next to each other. So these nuclei are really weird, and it seems that not all the invaginations accumulate poly-A, but those that do really, really, really get stuck in there. However, when we overexpress UPF1, that not only reduced NMD, or consistent with the fact that it reduced NMD fluorescent reporter protein expression and GAD45 and ARC1, it also accumulated the RNA within these invaginations. And so if you scroll down to slide 40, I quantified this and showed that the percentage of invaginations containing poly-A RNA were significantly reduced when you overexpress UPF1. So that kind of followed our hypothesis, or it was consistent with our hypothesis, that increasing NMD effectively reduces accumulation of RNA within these invaginations. So perhaps it is an overwhelming event. And so in the last couple of figures, we started taking steps to answer that question and determine the mechanism. So slide 41, how does tau limit NMD activity? Again, thinking back to our hypothesis, we thought that it was this tau-induced increase in RNA export. So similar to what we did previously, I genetically reduced expression of SVR and NXT1, which are the Drosophila homologs of nuclear export factors. And I found that in a tau background, this significantly increased clearance of GAD45 and ARC1 via NMD. And similarly, we saw this for the NMD fluorescent reporter protein. So there was less of this. So this suggested that genetically reducing RNA export increased the ability of NMD to clear RNA, and not only clear RNA, such as GAD45 and ARC1 and the fluorescent protein transcript, but also the protein. So it prevented downstream translation. And so we showed that genetic expression works really well, but that's not always the easiest nor the best strategy for humans. So we wanted to know, are there any agents out there that activate NMD, and do they work in the context of tauopathy? And And do they manipulate the NMD activity reporters that we're using? So there's not a lot of NMD activators. Nobody really looks at them. They're starting to gain a little bit more traction. There was a very, very recent paper that did look at some different NMD agents. But at the time when we were doing this, tranilast was recently shown to activate NMD in a Drosophila model of ALS. It is an anti-allergic drug. It does a lot of things. It's approved, but essentially right now, it's approved in Japan and South Korea for treatment of bronchial asthma. But it's been being looked as immunosuppressant and for very different things as well. But we use this, and we treated tau-induced Drosophila in control flies for 10 days. So from day 1 of adulthood to day 10, we added tranilast to the Drosophila food, and then measured at day 10 NMD fluorescent reporter protein expression, and found that it seems that there's reduced expression of this, so it seems to be activating NMD mediated clearance of this reporter protein, as well as of GAD45 and ARC1. So what they saw in Drosophila ALS models, we also see in Drosophila models of tauopathy. So moving on to some of the final slides, in slide 45, we showed that tranilast does indeed activate NMD. It doesn't matter again, so using counting tunnel-positive cells in the brains of tau-induced Drosophila, we found that tranilast-fed flies had significantly less neurodegeneration compared to vehicle-treated. And it did this without affecting tau levels. So it seems that, not only is genetic activation of NMD protective, but also pharmacological activation is. And finally in slide 46, we didn't do this previously, but we looked at neurodegeneration. Does this affect also health span, quote unquote health span? So we looked at locomotor activity in Drosophila, just basically counting how fast they can walk on a grid within 30 seconds, and doing that lots of times. So we found that tranilast-fed tau-induced Drosophila have increased locomotor activity at day 10. And possibly at day 20, suggesting that pharmacological activation of NMD improves health span in the context of tauopathy. So if you move to slide 47, what I'm going to try and summarize a little bit of what I've told you, and then how this kind of fits in things that we've also found in the lab. So I'm showing on the left is early tauopathies, so prodromal asymptomatic stages, and on the right is this age-related transition to the symptomatic or late-stage tauopathy. And so, as I mentioned, we see this early on. We see increased or deficits in NMD, potentially because there's increased RNA export based on our mechanistic studies. And this leads to increased translation. However, at this early stage, there is a sufficient amount of protein quality control. So you have normal neuronal function, so no neuronal death. However, when there is this age-related or tau-related changes to the protein quality control system, and late stages, it seems that these deficits in NMD become detrimental to the neuron, which is no longer able to keep up with the production of faulty RNA and toxic proteins. And so we propose, and hopefully I convinced you, that not necessarily tranilast, but NMD activators can help relieve the burden of this high load of RNA and this high load of toxic protein, and hopefully allow the neuron to keep up with this this overload at the step of protein quality control. And yeah, that concludes, at least for the paper, and I can take any questions, go over anything that I went too fast over, or yeah, if anybody has any questions. I'd just like to thank my lab and all the funding support.
Well, thank you so much for this great presentation. It's such an extensive and amazing work that you guys did. It's so impressive and also very important. So please flash your microphones if you have questions, anyone. And yeah, Dr. Shah, go ahead.
Yes, thank you so much, Dr. Zuniga. That was a wonderful, research. And my question in a clinical level, because we know that in an MRI of some of the patient, we saw the tau entanglement, but in the same time, they are not symptomatic, and it takes time that they want to show the symptoms. In the same time, your research just taught me about the microglia and the effect of the NF-kappa B in relation with the microglia and inflammation in the brain. I was just wondering if you have further information into this level for the, diagnosis. Thank you.
So I just want to clarify. So you're asking, so you see tau tangles early on, and then you're saying there's also inflammation early on at this stage, and whether that
Yeah, we are aware
has
of the toxicity of the, tau entanglement. In the same time, there was a research about the microglia and effect of the NF-kappa B in inflammation, and how we can just may suppress this. And I was just wondering, maybe you have any further thoughts around your research, in alignment with this type of information.
Okay. Hopefully I'm answering this right. But so in terms of, okay, so we see tau early on. We see these deficits of NMD, and you're saying like affecting inflammation. Yeah, so NMD also has been shown to be in the inflammatory pathway by acting in this viral defense. It's also shown to affect transposable elements, including copia, which is in Drosophila, and that can cause neuroinflammation. So I don't know how much, of effect. I don't know NMD's activity in microglia per se, but it's possible that, since we see these deficits early on, and perhaps they're also affecting that inflammatory pathway, that tau's affecting NMD early could lead to those symptoms, such as the neuroinflammation. I don't know if it's a high role in NF-kappa, sorry. But
Yes,
it's,
NF-kappa B.
yeah,
Exactly, doctor. That's why your research
okay.
was wonderful, and it just taught me about that. Not only in early detection, because we know that some of the patients they don't show the symptoms. Actually, they just have a normal life, and later on they might get some problems. But in the same time, for the drug development and whatever you just shared with us, that's why it just came to my mind. Actually, that was wonderful research. Thank you, doctor.
Yeah, thanks. Yeah, I appreciate it.
Does anyone want to go next? Are you sure? Then I'll go. I think I think it's really interesting that this this different levels of RNA are really important for this type of disease models. Do you think that this neurodegeneration so what do you think are the factors that like most important, like let's in translational, like to, I know you don't 100% know, but why do you think this happens in some individuals and it doesn't happen in others? If this is the mechanism, but what triggers it? Are you planning on studying that, or is there a way to do maybe RNA sequencing and make like a prediction based on the study?
Yeah, so sorry. So there's been a couple of studies that have kind of tried to look at NMD activity variability between individuals. So in a clinical basis, because the most interest has been in looking at NMD inhibitors so that truncated proteins that have some function can still be allowed to become proteins and function in some order. So like cystic fibrosis, a lot of people are looking at that. And they've noticed that some patients have variable activity of NMD. And so some patients will have more truncated proteins and some will have less. And so one of the things that it's so difficult about clinical trials, as I showed you with the failure rate, is picking the right patients and the right targets. So whenever I think of it, patients with Alzheimer's disease, we really need to have a very individualized approach. And so the best way to do this in this context would to be maybe to see patients with elevated levels. I don't know, maybe looking at plasma and CSF and see if you have lots of GAD45, you have a lot of these faulty RNA, whichever they may be, most prominent in humans. Maybe you would take plasma. Plasma would probably be the easiest, and do RNA-seq, and then look at which of these transcripts have some of the NMD triggering features. So do they have PTCs? Do they have intron-containing components of them? Do they have these weird abnormal structures that are normally targeted by NMD? That would be probably the most helpful if this was to go, we want to find a better NMD activator, and then if we took this to patients, that would be the most that would be the best way to approach it, is rather than just give everyone NMD activators. Well, it would be great if it worked for everyone. That's probably not the case. And so finding those patients based on plasma markers such as NMD target elevation would help not only find those individual variabilities and identify ultimately the patients that will benefit the most. So I think that answered.
I did
Yeah,
have.
thank you so much. Sorry.
Okay.
No, I just wanted to thank for
Oh,
this really great answer.
Yeah.
oh, okay. Yeah, I had a question. I'm curious, are the invaginations, is that something that's being caused, or is that, an individual variation? And if, and am I getting it right that it's the invagination that's effectively, trapping the RNA, and it's accumulating, and the NMD has to clear it, but it gets overwhelmed? Is there something? Oh, go
Yeah.
ahead.
Sorry. Finish your question. So sure.
Oh, yeah. Just to restate, is it something causing perhaps more invaginations, or something specific about them that trap the RNA, or?
Yeah. No, that's a good question. So to answer, we're still trying to figure that out. But so if you express tau in different model systems, you do frequently see these invaginations. However, they're not specific to neurons, they're not specific to tau. You'll see these, and they're frequently seen in cancer cells, as well as in aging cells. So in aging models such as Hutchinson, progeria, Guilford syndrome, you'll see tons of these invaginations. We see a lot more though versus postmortem brain tissue from Alzheimer's patients. When we looked at patients with the primary tauopathies, such as some nuclear palsy, you see tons of these invaginations. Like they're very, very wrinkly nuclei. But to answer the second part, we don't know. So we have a couple of theories. A lot of people ask, are the RNA just kind of stuck in there? Because it does look like that based on the fluorescent imaging. We don't know for sure because we haven't done any kinetic studies. So one of the things we would love to do next is to use culture system, use iPSC derived neurons expressing mutant forms of tau, and then do FRAP or to look at whether the RNA can shuttle in and out of those invaginations. So those are planned to kind of answer that question, but there's a really interesting idea where perhaps so neurons have a lot of long genes, and a lot of these intron containing genes are a lot longer, and so they can form these R loops during transcription. And a lot of these nuclear invaginations, as I kind of suggested, they terminate at sites of transcription. So at the nucleolus, you'll see them terminate there frequently. And so it's also possible that just recently thought of a theory was that R loops, while the mRNA is not only being transcribed, it's being exported through the nuclear pore. And inside these invaginations, you see actin, you see microtubules, you see tau, you see ribosomes, you see a lot of stuff in there. It could be maybe like the RNA could be pulling on and forming these invaginations. So we don't know yet if it's, this faulty RNA causing these invaginations to kind of be pulled inside, or if it's the other way around, to be decided.
Well, that's interesting. I've done some work with extracellular membranes. I haven't really studied nuclear membranes. But did you say that the tau could in fact be causing that? And is that like a surface curvature induced kind of effect?
Yeah, so the current hypothesis and idea is that tau will bind to filamentous actin, cause overstabilization and bundling of this actin, and it will essentially like poke into the nucleus, to the nuclear membrane. And it does this because actin is connected to the lamin nucleoskeleton, which I pointed out earlier in the slide, which is connected to the actin via the link complex. And so this causes the nucleus to invaginate. And tau also causes reduced lamin levels and most localization. So this kind of causes the nuclear membrane to be, the morphology to change, and for it to be very like foldable or just manipulated easily.
Oh, I see. So it at the skeleton level, the actin, it sort of gets its legs kicked out and just buckles.
Yeah.
In a sense. So, because I could see how invaginations of the appropriate size would dramatically reduce diffusion rates, and things would just get stuck. So, but when you upregulated the NMDA, it was able to get in and clear. So it's just it's sort of stuck but not walled off, or
right. At least that's what we think.
Fascinating. Thank you.
Yeah.
Some work that I've done in the past involved modeling like bacterial walls with peptides and seeing to what extent we can control the formation of pores. I would be suspicious that perhaps an antibody could be a kind of collaborator in this kind of event or something of that sort. And it makes me think of like actually recent advances in, quote unquote, peering inside of cells where they've had interesting abilities to differentiate between certain metabolic states using microdoppler radar. So that might be an avenue, at least for the kinetic situation, where there is kind of a dynamic metabolism and there's a lot of activity. You might be able to see significant differences and correlate them empirically. So then you could notice as an early warning system, or how early could you make that sort of assessment and to what extent. So that might be an interesting avenue.
Yeah, thank you.
Yeah, I posted the link in the group chat for the room, as well as your back channel.
Yeah, thank you.
Before
So
I forget, oh, go ahead, Eli. Go ahead.
just a quick mention, when Dr. Shah mentioned NF kappa beta, or NF kappa B, excuse me, I had to search tau and nonsense-mediated decay and COVID, and there was an interesting paper that came up out of the many results, and I put that in the room chat. I think that both Dr. Jha and Dr. Zuniga would be interested in it.
I had a
Thank
question.
you, Eli.
Hearing.
Dr. Zuniga, thank you for your amazing
Right.
presentation. It was
1 second.
very, oh, no worries.
Okay, I'm back. Sorry.
No worries. First, thank you for your presentation. It was very enlightening. Obviously, the slides were, I like the slides a lot. Did anyone else notice that on slide 32, it kind of looked like Mickey Mouse?
Oh,
I noticed that.
oh yeah.
I was curious about the mRNA expulsion mechanisms. It sounded like clearance failure was a consideration in all of this equation. And I was also curious about the survival rate of RNA outside of the cell in the blood. And given that COVID is some patients that are fortunate enough to have competent medical care are being diagnosed with Alzheimer's and other sorts of issues like that. I was curious if there was anything you could speak to for patients to perhaps, I mean, obviously understanding everyone is different, maybe a general recommendation for people who are having these sorts of symptoms in terms of treating themselves when there's a lack of clinicians that are able to treat them. Thank you.
So I just want to make sure I got this right. But your question, you're asking, what about the patients that are being diagnosed with Alzheimer's disease who have COVID? If that's related to this topic or just in general?
Well, I mean, that was the sort of the last part. I was
Yeah.
hoping we could go through the failure to clear the mRNA, or sorry, not the failure to clear the RNA, and how that's detectable in outside of fluorescing microscopy. Maybe there are markers, blood tests, or something to get.
So yeah, that was kind of what I was proposing. Could be a possibility in determining like individual differences. I don't know at this step. It's also, yeah, there's so many issues with plasma samples. So like how they're collected, how they're stored, that can affect RNA. So anybody who's worked with RNA knows that it's kind of finicky and it can be very hard to really quantify that, accurately quantify it. So that's the hardest part at the clinical step, is making sure that, every location, every patient sample is treated the right way, and that you're getting accurate measures. So, that's also a problem. So, as far as the neurological symptoms in patients that are being inaccurately or, called having Alzheimer's disease, I haven't followed too much with that. So I don't know if anybody else has kind of any thoughts, but in general, I think there's just so much new patients, new cases are coming up all the time. You follow patients, depending on, longitudinally, everything's just so new. It's kind of hard to classify these patients. And I mean, even now, Alzheimer's disease is being diagnosed more than it was previously. So, and the classification is changing all the time. So, I don't know where that's going to go in the future. It's probably going to change as we get more information, and if we get more better biomarkers, especially with the complications associated with COVID, that just makes it even harder.
Yeah, for sure. It is certainly a complex issue. Maybe I could focus the question. RNA clearance was a failure in this, well, a failure factor in this entire equation. Is there anything that can be done to increase perhaps the clearance, because that seems to be implicated in the mechanism of action for the dysfunction?
Other, do you mean other than targeting just getting more NMD machinery?
I'm not sure. I have no idea what the
Okay.
answer might be. That's why I'm asking the expert.
Yeah, so we think, I mean, based on what we showed, maybe, genetic overexpression, giving more NMD factors. So this would be genetic therapy. Obviously, that can be very difficult and complicated and expensive. So that's why we took the more pharmacological approach. The problem, yeah, I'm not going to go ahead and like recommend Tranlast by any means. We don't know how it's activating NMD, and so the next thing, if somebody takes over this project, is really to identify a direct NMD target. So I kind of mentioned in the discussion there are issues with that too, and that's probably why it's been really difficult for people to find that, is that each mechanism by which NMD targets its RNA and effectively clears it is different in different cell types and species. So it is very well studied. While it's very well studied in humans, different cell types are different, a little bit different, and so that makes it a lot more complicated. So then you go, is the drug getting where you need it to go, and is it going to the right cell type? What happens when it doesn't? So it is a lot more complicated, but it doesn't mean that strategy wouldn't work as a an additive, give them for the patients that need it, that show reduced NMD activity, maybe they'll benefit despite, somewhat side effects, if there are any. A lot of these, at least Tranlast, is very, very well tolerated and does cross the blood-brain barrier. So those are our ideas moving forward. They definitely need to be validated, and that's kind of the point of our study and why it was in Alzheimer's and dementia. It's really meant to say, okay, this worked in this model, now let's all go ahead and test it in vertebrae and human tauopathy, and see if you see similar things, and if these NMD activators also work there.
Amazing.
That's awesome.
Thank you so much.
It actually reminds me of the platform. Yesterday's speaker, if I'm not mistaken, had the organoid platform. That seems like a natural progression that seems to reduce the need for an entire animal. You can just have these very specific platforms and do these very precise kinds of setups. So I think Katerina could show some information about that that you could pass on to your team.
Yeah, we're working on those too. We have the iPSCs and the organoids. Working on it.
So I'm wondering if you've at all related the effects of the nonsense-mediated decay to.
Sorry, I don't know if my Wi-Fi went out.
We can hear you. Can you hear us?
One,
We
two,
can definitely hear you. Can you hear us,
three.
Gabriel?
Yeah, sorry, I missed that question.
Okay, well, I was just starting.
Oh,
so I was wondering if you had related either tau or the nonsense-mediated decay to the unfolded protein response, because that's like another level of intracellular stress that can lead to cell death.
We can't hear you. Well, it probably won't help if I'm talking.
is it related to the unfolded protein response?
Have you at all correlated or thought to relate any of this to the unfolded protein response, which is kind of another layer of intracellular stress?
Yeah. So we kind of. One of the things I always wanted to ask was, what are the consequences of this deficit in NMD? Because while RNA toxicity could be something, it's not really something that people have shown in tauopathy, although what's really validated is the protein toxicity. So I wanted to see if it affects the proteome. And so I have data that we're about to submit, and where we see elevated translation, global translation rates. And so it seems like there's a lot more RNA protein that's being made in the context of tauopathy. And so it could be they've or previously, I can't remember when, it was quite a while in the early 2010 2011, the Feeney lab showed that the unfolded protein response is activated, suggesting that, there's this, like you said, this cellular stress going on in the tau flies, and maybe that's why you get these protein aggregates and tauopathies, because the protein quality control system is not working. And I think, and what we're trying to throw out there is that there are these deficits in NMD. You're getting a lot more RNA, but you're also getting a lot more protein. And while I haven't quite yet shown that this is quote unquote bad protein, it could also just be this massive amount of overload of protein that's occurring. And because there's already deficits in protein quality control, as that conclusion slide kind of suggested, that these massive amounts of protein are contributing to the proteome. And that's, this cascade that's leading into this activation of the unfolded protein response that's not quite cutting it. The neuron can't keep up with it at that point.
I had a caution, wild speculation ahead. I look at the tranilast, and I'm thinking, from work I've done on membranes, this is molecular dynamics level modeling the membranes and things in them. It looks to me like that, if that has any effect on the membrane, it would be in essence membrane stiffening. And I'm wondering if it's not known about how it activates MDA in this case. If it's not a specific MDA interaction, you said it does many things, so that kind of indicates some non-specificity. But if it acts on the membrane, and in essence, by stiffening the membrane, it would open the invaginations more, which would increase effective diffusion, and that could possibly indirectly allow the MDA to clear more effectively. I'm wondering what is known about the interaction and how it would activate MDA, and what's known about its effects on membranes in general.
Yeah. So nobody's really looked at it, at least to my knowledge, looking at the nuclear membrane. It's definitely, like what you said, is definitely possible. That was one thing we thought to do, was to look at the invaginations and internalized fed flies. But yeah, one of the theories is that because it's this, it modifies the inflammatory response and maybe the viral response, that this modification is somehow, like I mentioned, NMD also plays a role in that viral response, responding to transposable elements and double-stranded RNA, that indirectly, through the inflammatory pathway, it activates NMD, but that hasn't really been validated or confirmed. But that's the theory.
Well, thank you. This is really fascinating material.
So I have one question that Victoria asked me to ask, and she had to leave, but I promised her to get back to him.
Yeah.
do you have a book that you really recommend, either in your field or in general, like not like nonfiction book that kind of was important for you?
It's a good question. I don't really have a recommendation. Sorry. I'm trying to think. It's been a lot of just reading papers lately.
Oh, so maybe a paper suggestion, like maybe top 3 papers or some sort of author that you're like, okay, this is the stuff that really got me going recently.
Yeah. So, I've been reading, who does similar work. Not sure if I'm saying his name, but Joe Abisamra's group in Florida, they do a lot of similar stuff, looking at protein translation. And they did it in tau-induced mice, actually. So they also take a similar approach. And then Schulman group and Baylor, they've reported also that there are these splicing defects, that, so if anybody's else familiar with NMD, it's really tightly connected with alternative splicing. So their group showed that there's this massive change, in splicing patterns, and that there's intron retention. And as I mentioned, and as our reporter mentions, that you have these introns, that they're targets of NMD. So that kind of follows with our idea. And so I've been following these two groups and kind of seeing where their pathway and our pathway kind of lines up. But that's probably not as interesting as a good book. Sorry.
Thank you. Yeah,
No, no,
thanks.
I think that's great. Like, I prefer to hear about the papers because they're like advanced. Books are like, yeah, I wrote this book 10 years ago, didn't update it. Chapter 2 is still spelt wrong. Deal with it. It's kind of that silliness. But the papers really have to go through sometimes the gauntlet, and I find that it's much more interesting, especially even if it's an amateur person looking at something. I'd recommend something like Rewordify, because I know at least for the first time through, even just trying to read out some of these molecular names, trying to remember how you say it according to some Latin convention can be kind of tedious. So I recommend to folks in the audience, perhaps the general public, to look at something like rewordify.com, I think it is, if I'm not mistaken. So that takes like complex legalese or complex scientific flesh Kincaid level 25 writing to something that, a fifth grader could understand. Although not the fifth graders from that show, because those children know too much. But yeah.
No, that's a good reference. Yeah.
That's really great advice. Thank you, Eric and Gabrielle. I think, yeah, that's really cool. Does anyone have any questions in the audience? Maybe. Ram asked, what are biosimilars and difference between biosimilar and biogenetics? I'm not sure. Do
I
you?
think you mean biogenics.
Okay.
But I'm not sure what the question pertains to. Sounds like a molecular dynamics question.
I don't know if I can answer that one. Sorry.
Yeah.
So if I remember correctly, biosimilars is a term that's used with regard to approved drugs or approved biologicals. It's kind of related to the difference between brand name drugs and generics. I'm not sure the pertinence to the topic here, but that's what comes to mind.
So, do you think, since it's a neurodegeneration, did you ever try to also look, or are you planning to also look into Alzheimer, ALS? I'm so sorry, into ALS. If this is maybe a more general mechanism that maybe applies to, different disorders, and not just in the context of, Alzheimer's.
Yeah, so the paper we found Trinilaz, they actually did just that. So they looked at they used a Drosophila model of ALS, and then they also used iPSC derived neurons, I believe, from ALS patients, and found similar things. So that's ALS is notoriously an RNAopathy, and so they did find that there are deficits in NMD. Similarly, however, there are differences in the theory of how this is happening. So ALS, you do see invaginations, but they don't see this excess RNA export. Rather, they see these tau aggregates with nuclear pores. So there is aberrant nucleocytoplasmic export, but it's less. It's not causing this accumulation of RNA and this overload. They actually think it's affecting translation, which, since NMD is a co-translational decay, that is effectively inhibiting NMD. So while it seems that deficits in NMD are a conserved feature of neurodegeneration, we don't think that the mechanisms are similar, and the downstream effects. While in ALS, it's, as I kind of mentioned, it's causing this RNA level toxicity, whereas I think in tauopathy, it's not only causing toxicity at the RNA level, but also at the protein level.
Oh, interesting. Yeah, that's really interesting to know. Um, but yeah, what about, so just general aging? Is this mechanism also involved in just, general aging, like a aberrant mechanism in this pathway? That would be also interesting, I think, or maybe there's already somebody looking at this.
Yeah, so we showed we showed that there's a trend in an age-related decline in NMD. However, there was a paper in 2017 that did this in C. elegans, and they found that there is an age-related decline in NMD activity, which, is just consistent with the whole aging theory that protein quality control decreases and RNA quality control decreases. But they were showing that NMD specifically decreases with age. And as I kind of mentioned, the Hutchinson progeria Guilford syndrome, which is a premature aging syndrome, they have lots and lots of invaginations. So I don't recall, probably somebody did this, but I don't recall if the invaginations get worse with age. But there is definitely an age-related factor. So it's an age-related factor in controls that's enhanced in tauopathy. But beyond those papers by that specific group that looked at age-related decline in NMD, there hasn't been a ton looking at its mechanism in aging, and it would be really interesting to look a little bit further in that, maybe in a mammalian model of aging, or in cell culture, or something like that.
Yeah, I agree. So, do you think that, like, so basically it's just basically the control mechanism to check if proteins are faulty or not, are involved, and not like maybe that some type of non-coding DNA is kind of also disrupting this mechanism in some form. But it's really hard to know, right? In aging,
Yeah.
all stuff happens at the same time, so it would really be hard to know. Are you planning on studying, like, is there any lifestyle type of study, like sleep, that is affecting this, like a study that shows that it's affecting this mechanism of poor um, um, food intake or some kind of chronic stress? Is there
Yeah.
that? Yeah, like I don't know if you.
I don't think or to my knowledge, nobody's looked at that, but we know that that's a factor in Alzheimer's disease and in tauopathies, that there's sleep problems, sleep changes in sleep pattern, and then we have some data that we have a model of tau spread that we're working on, and there seems to be this circadian rhythm that is really weird in tau, and this affects the spread, but we haven't really pieced together whether that's related to other factors. But as far as I don't think anybody's looked at different kind of stresses on NMD. This is still kind of new. So the mechanism of NMD has been well studied, but looking at it in disease pathways such as in tau and ALS, these are relatively new in the last 2015 onward. So I'm not familiar with any papers, but I would be really interested to see if like you said, any kind of just stressors that we all have every day affect NMD activity.
So just a quick mention. It occurred to me to search NMD and cytokine response, and it appears that at least looking at titles of a few papers, that there's some relationship between sensitization to cytokine driven cell death when NMD is upregulated, and this is just kind of the straightforward prediction that NMD would be involved in innate immunity to viral infection, and so that would polarize towards apoptosis. And of course, something like that would also, contribute to driving aging and sensitivity to other things and so on.
Yeah, thank you so much. I think that means you have a lot of research ahead of you. You can probably keep working on this for many, many years, with different stress factors, lifestyle.
Mm-hmm.
Yeah, it's really interesting because of that, because it's still unknown. So congratulations again for your great work and for this paper. And thank you so much for taking the time to present your work here and to answer all these questions. I think this was really interesting and it was a great conversation. So thank you, Gabrielle.
Yeah, thank you all for attending, and I really appreciate the questions and the feedback and all the advice. So thank you.
And
Thanks, Gabrielle. +1 to everything Katerina just said.
and
Yeah, thanks, Gabrielle.
and thanks everyone for coming and participating in the discussion, for asking great questions. And I hope to hear you all back soon. And maybe Gabrielle, once you have more data that you can talk about, maybe you come back and present it to us. That would be really wonderful in the future.
Sounds good. Thank you.
Thank you. And if you like discussions like this, please join the club, Science Society, if you would like. And we have a room tomorrow earlier at 5 p.m. EST. It's Dr. Henry from MIT, and he will talk about his development of thermophotovoltaic system that reached efficiency of 40%. So I think that's really exciting in the context of um, climate change. So yeah, come back if you would like. And Gabrielle, thank you, and always feel that you're always welcome to come back. Maybe to even just take part in the discussion when guest speakers are here. And thank you so much again, and thank you everyone. Okay, let's close the room in 3, 2, 1. Bye everyone.